Hello everyone, welcome back to my channel. In this video lecture, we will cover our next topic of the chapter which we were learning in the previous video, that is the double fertilization. So basically double fertilization होता क्या है? Syngamy and triple fusion is known as double fertilization. Then the pollen tube releases two male gametes in the embryo sac. One of the male gamete fuse with the egg to form diploid zygote. This is called as syngamy or called generative fertilization. Then the second male gamete fuse with two polar nuclei. This produces a triploid primary endosperm nucleus, and this is called as triple fusion and also called as vegetative fertilization. In an embryo sac, there occur two sexual fusion, one in syngamy and other in triple fusion. Then together this phenomenon is called as double fusion, double fertilization. क्या होता है? pollen tube enters into synergates. The pollen tube releases the two male gametes in the cytoplasm of the synergate. That syngamy occurs, then triple fusion occurs जहाँ पे three haploid nucleus fuse. Okay. This diagram is showing the triple fusion. Then post fertilization events, structure and events. Endosperm. Endosperm is the nutritive tissue formed from vegetative fertilization. Endosperm is meant for nurturing the embryo. It is generally triploid, and the endosperm may show the effects of genes from the male gametes, and this phenomenon is called as xenia. ठीक है? फिर the nuclear endosperm. Primary endosperm nucleus forms a large number of free nuclei by repeated mitosis. Central vacuole is then formed which pushes the large multinucleate cytoplasm to the periphery. Then the second cellular endosperm wall is formed after division of primary endosperm nucleus. Therefore, the endosperm is cellular from the beginning. Haloboid endosperm. First division produces two cells. Free nuclear division may occur within the cells. Okay. These are non-persistent embryo in some seeds and perisperm till germination. अब फंक्शन क्या है एंडोस्पर्म के? फंक्शंस ऑफ एंडोस्पर्म आर इन प्लांट्स विद एल्ब्यूमिनस सीड्स. एल्ब्यूमिनस सीड्स क्या होती हैं हम आगे पढ़ेंगे. In plants like albuminous seeds, nutrients in the endosperm help in early seedling growth. Then endosperm provide nutrition to the developing embryo. Then the liquid endosperm of coconut contains oxygen, cytokinins and Ga and includes cytokinases when added to basic nutrient medium. Coconut milk can also be used to induce the differentiation of embryo and plantlets from various tissues. Zeatin is the very potent cytokinin. It is extracted from young endosperm of maize. Okay. Then embryo formation, embryogeny. It is the development of mature embryo from zygote. Then early development produces a proembryo which is axial symmetry. Dicot embryogeny. Zygote divides into two unequal cells. Larger suspensor cell towards micropyle and a smaller embryo cell towards antipodal region. Suspensor cell undergoes transverse division forming 6 to 10 suspensors. Then the first cell of the suspensor is called as haustromium and last cell is called as hypophysis. It forms radicle. Embryo cell divides twice. ठीक है? Vertically and सबसे पहले vertically फिर transversely to produce two tiered 8 cell called embryo. Then it is initially globular. It becomes heart shaped later and finally takes into a typical shape. ठीक है? फिर monocot embryo में क्या होता है? The zygote undergoes transverse division to produce a vascular suspensor cell facing the micropyle end and the embryo cell facing the chaliceal end. Where the embryo cell undergoes another transverse division to form a terminal and a middle cell. Terminal cell undergoes vertical as well as transverse division to form globular embryo. फिर ये form large cotyledons and a plumule. सबसे मेन बात डाइकॉट में cotyledons नहीं होते हैं, मोनोकॉट में होते हैं. As the cotyledon grows in monocot embryos, it pushes the plumule to one side, and in some grasses the remnants of the second cotyledons may be seen. It is known as epiblast. Single cotyledons of monocot is known as scutellum. It is shaped like a shield and appear as terminals. Then this is the most important topic, transformation of parts of flowers. Before fertilization, calyx, corolla, endoecium, style, stigma are present, and after fertilization they wither off. And ovary, before fertilization ovary becomes fruit, after fertilization ovary wall becomes pericarp, ovule becomes seeds, integument becomes seed codes, outer integument become testa, inner integuments become tegmen, micropyle become remains micropylorate, funicle becomes the stalk of the seed, nucleus become perisperm, egg cell becomes zygote, and synagogues disintegrate and disappear. Then formation of seed and fruit. Fruit is basically the ripened ovary or fertilized ovary. Wall of the ovary forms fleshy or dry fruit wall called pericarp. Fleshy fruit or pericarp has 3 layers: epicarp, mesocarp and endocarp. It is the covering of fruit that develops from ovary wall. Then it is the part of fruit and is dry or fleshy. It is protective covering and provides nutrition to the seeds. Ripened ovule are known as seed, and integuments of ovule form seed codes. Integument becomes seed coat, micropyle becomes seed pore, ovules become seed, ovary become fruit, and wall of the ovary become the pericarp. As types of third integument, in some cases like lychee, it provides an additional covering of seed. Then funicle forms seed stalk. Larger seeds are of those double coconut which are bilobed, and each seed has weight of 6 kg. Then depending upon the presence or absence of endosperm, the seeds are classified as non-endospermic or exalbuminous or endospermic or albuminous. Then non-endospermic or exalbuminous में food stored in endosperm is completely used up before the developing embryo. Example, gram, pea or bean orchids. And endospermic or albuminous are endosperm grow vigorously and is not used by the developing embryo. Then what is the importance of seed? Evolutionary achievement. It provides protection to embryo. Seeds have sufficient food reserve. Seed can colonize and populate new areas and spread and propagate. Germination and sowing of seed by humans gave rise to agriculture. Then seed viability. It is the period of time for which the seed retain the ability to germinate. Then seed viability is determined genetically. Then first, respiring seeds then corollas, triphenyl tetrazone, chloride into pink, triphenyl formazone. Then first is apomixis. It is the formation of new individual by asexual method which mimics sexual reproduction including seed formation but do not involve fusion of gametes or sex cells. Then normal type of sexual reproduction having 2 regular features, meiosis and fertilization, is called amphimixis, and the organism reproducing through apomixis is called apomycetes. Then it occurs by following method: formation of asexual seeds if the embryo develops directly with gametic fusion. Then sporophytic budding if embryo develops from diploid cell of nucellus or integuments. Then terathogenesis, development of new individual from a single cell gamete without fusion with another gamete. Then depending on the ploidy, these are of 2 types: parthenogenesis haploid and parthenogenesis diploid. In haploid parthenogenesis, embryo sac and eggs are haploid, and in diploid parthenogenesis, embryo sac as it contains egg, it is diploid and it undergoes parthenogenesis and forms diploid embryo. Then apogamy, Formation of sporophyte or embryo directly from the cells of gametophyte. In higher plants, only diploid apogamy is successful, that is, gametophytic cell forming the sporophyte is diploid. Then polyembryony, phenomena of having more than one embryo is called as polyembryony. Occurrence of polyembryony due to fertilization of more than one egg cell is called as simple polyembryony. Then formation of additional embryos can also occur from different parts of ovule like synagids, antipodal, nucellus, etc. Then polyembryony is more commonly seen in gymnosperm than in angiosperm. Polyembryony can be false or true embryony. Then parthenocarpy, it is the formation of fruit without an event of fertilization. Parthenocarpic fruits are seedless. Genetic parthenocarpy is due to genetic alteration caused by mutation. Environmental low temperature, frost and fog can induce parthenocarpy. Chemically induced are the spray or paste of auxins and gibberellins, that are the hormones. Then importance क्या है इनकी? They do not contain seed which have to be removed before eating. Fruits can be developed inside the greenhouse where pollinators are not available. Then it quicker the food processing. So this much for today's video. Hope you liked it and understood the chapter, understood the main topics. If you are having any doubt, please do let me know in the comment section. Thank you for watching my videos. Stay safe, stay learning and stay growing. Thank you and don't forget to subscribe my channel. Till then, bye bye.